Marcus is a master beekeeper, and he is the U.S. Beekeeping Operations Manager for Desert Creek Apiary in Texas. And he's going to talk to us tonight about honeybee nutrition, biology, physiology, and winter readiness. And Marcus, if you could talk to us a little bit about what your job entails and how you handle that before you start, that would be great.
Okay. Well, yes, I appreciate you all inviting me. Thank you. My name is Marcus Hill, and I am the Beekeeping Operations Manager for Desert Creek Honey, Desert Creek Pollination, and Desert Creek Bulk Bees. So basically, we do our honey production in Texas and the Dakotas. We manage about 70 to 80,000 hives in the almonds, and we also sell nukes packages, full hives, and do splits for other commercial beekeepers in Texas as part of the bulk bees program. So if it involves the living organism of a honeybee, it's essentially under my oversight. So that's my current role. A little bit of background. I was a beekeeper in North Carolina for 12 years or so. Started out as a hobbyist, and well, my beekeeping career first began about 20 years ago on the big island of Hawaii, where I went from knowing nothing about bees to doing it about 16 hours a day, 6 days a week, rearing hundreds of thousands of queens a year. And then I moved back to the mainland, and my wife and I relocated down to North Carolina. I took a research tech position at NC State, helping different postdocs and other researchers at the Tarpey lab with their work. And then I took a position as a research scientist doing contract research for plant protection product registration. So I built a business unit called Bee Health and Management Solutions, which helped enable the Environmental Protection Agency to get viable data, replicable studies in order to best label different plant protection products as far as bee exposure and what effects they might have on honeybees, as well as what residues there may be in pollen or nectar coming back from the different plants. Also spent a number of years with my own commercial operation, a couple thousand hives, and right now am at least 10x that with my current position at any given moment. But I wanted to take some time today and kind of go over some stuff, basic bee biology and physiology, as well as how it impacts both winter readiness and the following spring. Some of this might be old news for everyone, but hopefully there's some tidbits in here that are educational. And if anyone has any questions, I'm happy to take questions as well. Let's see. Can I share my screen? Let's see.
Yes, you've been made a so you can share.
One of our sister companies is the Bee Supply, so thus the logo. Another one of our sister companies is Commercial Bee Supply. So we are in all things honeybee, both from hardware and hiveware to honey production to pollination to queens and all the above. So I entitled this Bees, Nutrition, and Winter Readiness. And being ready for 2025 actually pretty much started at least in August. So I hope everyone has treated for mites by now and is, if needed, heavily feeding their bees. A little bit of information on honey and wax. It takes about 6 grams of sugar for every gram of wax and about 150,000 miles of foraging flight per pound of wax. It takes 6 milligrams of sugar per day per nectar forager for them to have the energy to fly. Now, about 40 grams of wax can hold 4 pounds of honey, And honey is nectar less than 18.6% moisture, but in the marketplace, packers are really looking for about 16.5. So a newly built comb consists of recycled comb, brood cappings, and new wax is created when all the comb is filled and the bees are satiated, but how full those cells are doesn't really matter. If they've got nectar in them, they consider it filled. So during a flow, they'll keep building. Pollen, bee bread, and brood food. As we know, pollen is the protein source for bees. It can take up to 12 weeks for a colony to recover from a protein dearth. Foraging is regulated by colony demand, physical inspection of the open brood cells and cells near open brood as the foragers unload their pollen. Honeybee foragers unload their own pollen, unlike pollen foragers rather unload their own pollen, unlike nectar foragers, which pass that off to others and go back out to gather more nectar. So they assess the brood level and the larval age via the brood pheromone. And pollen foraging may also be influenced by trophylactic contact. So in the pollen are the essential amino acids, the largest requirement being leucine, isoleucine, and valine, but there are 10 amino acids that are essential for honeybee growth. Not all pollen is digestible. About 23% of it is not. And pollen is also the source for vital phytonutrients like omega-3s and omega-6s. And just a helpful little tidbit, another fantastic source of your amino acids and phytonutrients are bananas. So if you're feeding your bees now, for example, which you should be in North Carolina, if you don't have at least 65 pounds roughly, then you want to be feeding. But I would recommend taking a banana, slicing it lengthwise, and putting it on top of your brood chamber. Leave the peel on there. It has a lot of nutritional value too, and you'll have longer lived, healthier bees as a result. So when you feed them, throw half a banana on there too. It also helps combat chalk brood. There aren't really any studies that show that, but there's a lot of beekeepers that see that. So pollen and bee bread account for about 5% of all protein feedings to brood. You might notice as you're looking in the hive and the pollen foragers are depositing the pollen they've collected in cells near the brood. They're walking across the open brood, and you can't help but have pollen fall off of the corbicula into those developing brood cells. So developing queens are fed exclusively royal jelly, and it's the absence of that pollen which allows them to develop to full reproductive capacity, which is why the queen cell is essentially an umbrella. So the bees can walk across that, no pollen falls off their corbicula and inhibits her complete development. So nurse bees, they consume more pollen that's roughly 1 to 3 days old than they do older stores. They prefer fresh pollen. They consume the majority of pollen and convert it to royal jelly. We'll dive a little bit into queens, 1,500 eggs per day. Intercolonial genetic diversity impacts colony survival. Essentially, all that means is the more genetically diverse or the more drones this queen is mated with, the more tools that hive has in its genetic Swiss Army knife to survive what they're facing. So they're polyandrous, multiple mating flights, and 1 to 40 partners, with an average of 12 in the continental US. In Hawaii, there's an average of about 40 patrolines in a queen. Queen quality is determined by a few different factors: the age of the queen herself, as well as the age of the larvae used to raise the queen, which is likely one of the reasons why supersedure queens are of lower quality and have less capacity for that hive to actually overwinter well, because you need a queen that's essentially reared from a larvae that's just hatched. You don't want to wait too many days beyond that, because that just pollen's going to be falling into that normal brood cell before they or you who's grafting take that larvae out, put it in a cell cup, for example. And of course, the number of drones she's mated with impacts the quality, as well as the sperm viability, which does decrease over time. So drones, in brief, they're haploid, meaning they have one set of chromosomes. Honeybees are haploid diploid. Female honeybee is from a fertilized egg, and a unfertilized egg becomes a drone. There are at times what are called diploid drones, which are a fertilized egg that became a drone, but they are cannibalized within a few hours of hatching. The main role for a drone is one-time mating with a queen. They have less than a 1 in 1,000 chance, so the odds aren't that good, but they make multiple trips to the congregation area per afternoon and about 30 minutes per flight. The best drones are about 10 to 21 days old. If they're less than 10 days old, they're too immature, and if they're over 3 weeks old, their semen's too viscous. Just a little bit of information on the queen glands. I won't get into them, all of them too much, but the most important for our talk now being the queen mandibular pheromone, which attracts mates, lets the colony know that they're queen right, ensures the retinue response. It attracts the young bees that serve as a retinue, and also moderates juvenile hormone synthesis in young bees. So as the juvenile hormone levels rise and vitellogenin levels fall, bees age, and they go on to become middle-aged bees and then forager bees as they become more and more separated from that QMP. The queen retinue pheromone also aids in retinue response and inhibits worker ovary development. It's a blend of different pheromones that are not produced in the mandibular glands. So her quality and your colony's viability are inextricable. Queen failure is a leading cause of winter mortality, and queen events like supersedure increase the risk of that mortality, as I mentioned earlier, possibly due to lower quality queens like those reared from older larvae. The queen's promiscuity lowers disease intensity, for example, Viromorpha serrani, which is what is most commonly known as Nosema serrani, but it was misclassified. But probably nobody's going to change their label on like Nosevit or something and call it Viromorphizit. But for the sake of accuracy, it's actually Viromorpha apis or Viromorpha serrani. Queen promiscuity also heightens colony strength, decreases the likelihood of diploid drones, and beneficially impacts the queen's physiology, like her pheromone profiles, which then impact the retinue response. And intracolonial genetic diversity, which is fostered by that queen promiscuity, as I mentioned earlier, serves to provide more genetic tools at the colony's disposal. A less diverse colony has a far greater risk of dying over winter, but that genetic diversity doesn't indicate whether or not they'll be able to thermoregulate well. It's another reason that they die out. The age of the queen, both chronological as well as the age of the larvae from which she's reared, also greatly impacts the colony viability. As mentioned, the sperm viability decreases over time. Queens reared from young larvae are larger, have more stored sperm, they mate with more drones, and have more productive offspring. The colonies headed by young queens have lower winter mortality risk. And it amazes me the variation in terms of commercially available queens. Some were 84% viable, plus or minus 13. Some as low as 20.5% sperm viability. Now, with some of that impacted in shipping, likely temperature can certainly fry sperm in a queen. So that's something to be mindful of too. The average number of stored sperm in a queen is about 4 to 5 million. And as I mentioned earlier on, in the continental US, it's about 12 patrolines on average, though some studies indicate 17 or plus or minus. And the queen, of course, is what lays the eggs, and it become your workers or your drones, or another queen. But the survival of that brood is dependent on a number of things, including pollen dearth or poor nutrition as a larvae and pupae, which can lead to early capping, early foraging, and early mortality. It's a delayed feedback loop. So if you have a lack of protein in the bees that are developing now, for example, and those nurse bees come out with a less well-developed hypopharyngeal gland, then the proteinaceous secretion, royal jelly that they feed to the developing larvae that they're tasked with caring for as nurse bees, aren't going to be as nutritionally sound either, and their hypopharyngeal gland is going to be even worse off, and it just is a downward spiral. That delayed feedback loop also includes the colony age and task structure. So the nurse bees move on to older bee tasks more quickly, and eventually become foragers, a little too early, and it just, as I said, creates a downward spiral. Inadequate nutrition and / attention to future generations of brood is also a direct result of poor nutrition. So nurse bees preferentially allocate protein to older larvae at the expense of younger larvae. They're going to keep investing what they've invested the most in. Dearths can be caused by lack of available foraging, lack of foragers, or just the inability to forage, like rain. Cannibalism, as I mentioned before, is a fairly common thing, especially in dearths. But simply moving or repositioning a frame can lead to cannibalism of developing larvae, as well as can the distance from accessible pollen that's 24 to 72 hours old. Nurse bees don't really go to it beyond 7 centimeters away. So that's why you'll find the pollen foragers and the pollen kind of tightly packed around that pool of royal jelly with a larvae floating on it. So eggs and middle-aged larvae are the most likely cannibalized. And as I mentioned earlier, diploid cannibalism results from a homozygosity at the sex allele, which yields diploid drones, and they're cannibalized within 6 hours of the eggs hatching. Queen-right colonies under normal conditions 48 years ago were found to have brood mortality at 20 to 25% in the spring, 10 to 20% in the summer, and up to half in the autumn. So it's very important in terms of moving into the diatomaceous bee production that the availability of protein, the essential amino acids, is abundant. I don't want to jump ahead of myself. I'll get into that a little bit more later, but a high proportion in this study in 76 was cannibalized typically towards the center of the nest. So we'll focus briefly on the hypopharyngeal gland, which secretes the proteinaceous royal jelly. It's most active in young bees, those that are in contact with open brood. It's plastic, meaning it's functionally flexible. So if an older bee has its vitellogenin level raised, that hypopharyngeal gland can return to function. Juvenile hormone level increases, which In turn, the gland shrinks, and the development of enzymatic activity for sucrose hydrolysis by forager-aged bees begins. The shrinking of the gland and developmental inhibition is also evident with plant protection products like protease inhibitors, and as well as varroa mite infestations. Even just 1 to 3 mites can inhibit the development of that hypopharyngeal gland. So I'm going to hammer the importance of treating for mites in a timely manner, especially as you're working towards having your bees ready for winter, having your winter bees, which were eggs that were laid in August, September, and is probably petering out now. So I would highly recommend, if you have a time machine and did not treat in August for mites, you jump back and do that. So an adequate number of well-nourished nurse bees is needed in the time preceding diatomeous production. And aside from ensuring that they have the proteins that they need, as well as the carbohydrates and phytonutrients, ensuring that your varroa mite levels are low. And my basic rule of thumb is, if I have 2 mites per 100 bees, I'm treating. So, and then I would do a little dive into nurse bees. Welcome to the world, and I get to work. They're developmentally immature at emergence. They spend the first few days cleaning cells, grooming, and resting. The proteinaceous jelly is needed at emergence for their developmental immaturities and physiological evolutions to occur. So they're fed royal jelly when they first come out, and they're able to develop that last little hurdle into being a full-fledged nurse bee after they've emerged. And they sleep in proximity to the brood nest and in cells. You'll see them, tucked in a cell, looking all snug as a bee in a cell. And they differentiate very little between day and night. At 4 to 12 days old, they feed jelly from the hypopharyngeal gland to the young, to workers, and the queen. They serve as the queen retinue, and they regulate her egg laying by their feeding rate. They also act as a vehicle for her queen mandibular pheromone. They help regulate pollen foraging based on the richness of the royal jelly that they're producing. And they also feed adult bees, though not necessarily foragers, about 1 time per hour, and handle an equivalent of 2 to 3 larvae during their nursing lifetime. So they've got a lot on their plate from emergence through the rest of their life. Pollen digestion takes from a few hours to a full day. There was a study done where 50% of radioactive tracers attached to injected proteins were found in the hypopharyngeal glands after 6 hours. I just found that interesting. Most nurse bees eat pollen within 3 to 4 centimeters of the brood area. And as I mentioned earlier, they don't respond to pollen that's beyond 7 centimeters away. So it can be a good idea when you're setting up your hive, if you have, the bees provide a great template. So you have like honey, nectar, pollen, eggs, open larvae, cap brood. And if you set your hive up that way, your outer frames, honey, nectar, pollen, open brood, so that the bees really just have to turn around to access the pollen. And then the cap brood, as it gets towards the middle, the bees provide a great template for how to set up your boxes by how they set up their frames. Moving on to middle-aged bees, those are bees that are about 12 to 21 days old. Their vitellogenin levels are still high, and vitellogenin is essentially the fountain of youth for honeybees and maybe for people, so eat more egg whites. The middle-aged bees are engaged in nest building and maintenance. They're cementing bees, guard bees, fanning bees, water carriers, and they move on to becoming nectar receivers as their juvenile hormone levels rise and their vitellogenin levels fall. And they receive that nectar and allocate it to different cells. nectar receiving and building aren't typically performed by the same middle-aged bee. So there is some division of task structure there too. And about 1% of middle-aged bees serve as undertakers, dragging out bees that died in the hive. Middle-aged bees are recruited to being foragers as receivers. They are forager ready, but they make that leap into, the suicide mission of beating your wings to death until you've brought back all the nectar that you possibly can in a relatively short lifespan. But as the time increases between when they're receiving nectar and contact increases with waggle dancers instead of nectar foragers seeking relief, those middle-aged bees feed their vitilogenin back to the hive, which essentially is them, saying, I'm going to go ahead and give up life in order to gather as much resources as possible for the hive. But that vitilogenin is the currency for the civilization within a honeybee hive. The proximity to the nest during sleep also decreases in middle-aged bees, and day-night differentiation increases. And they move on to foragers, which fly an average of 21.5 kilometers per day. As mentioned, they require 6 milligrams of sugar, carbohydrates per day for flight alone. And they're fed about 2.25 times per hour, though not specifically by nurses. Ethyl oleate, which is a primer pheromone produced by adult workers, can delay the onset of foraging. And those foragers, they get very little sleep in comparison to your nurse bees or middle-aged bees. They'll sleep about 13 to 17% of the time during the day and maybe half the night. But when there's foraging available, they don't sleep, and they don't sleep in the cells. And as I mentioned, workers feed their vitilogenin back to the colony before the foraging stage. Their hypopharyngeal glands atrophy, but as I mentioned, they are plastic. If they are fed back vitilogenin, they can return to function with that vitilogenin level increase. And when they fed that vitilogenin back to the hive and become a forager, their lifespan is shortened to under 5 to 6 weeks at the height of foraging. Moving on a little bit to what I said I was going to hammer quite a bit, varroa mites. The best mite's a dead mite. The economic thresholds changed quite a bit. 10 to 12% infestation rate was considered the economic threshold in '99, but proactive treatment should be used to keep mites below the threshold and to undermine viral infection rates and comorbidity caused by the varroa vectoring diseases, as well as the protease inhibitor like hypopharyngeal gland impediment to development that varroa mites cause. So if you haven't killed them yet, kill them now. I do want to dive a little bit into propolis, which gets a bad rap, but is a major part of the honeybee's immune system. So it's collected mainly May to November. Common sources are poplar, pine, birch, alder, elm, beech, horse chestnut. It can take a long time, from 15 minutes to 7 hours to unload. If you ever had propolis on your fingers, you're aware it can take quite a long time to get off. It takes 7 minutes to load the corbicula, but could also take up to an hour. It's an external immune defense, which enables them as an organism, individual, as opposed to the superorganism of a hive. It enables them to have less immune system investment. Though the sources of propolis vary the efficacy, propolis inhibits the development of microbes. It also has antimicrobial properties and benefits in terms of battling chalk brood, American foul brood, and even inhibiting the growth of varroa mites. It also inhibits it increases wax moth larvae mortality and can reduce their metabolic rate in the larvae in adults. So I try to advocate, if at all possible, on a large scale, on a commercial operation, it's not as feasible. But if you have a few hives in your backyard, take one of those plastic queen excluders and cut it into subsections and staple it to the walls on the inside of the hive, and they'll fill those cracks with propolis, and you can peel that off and help to kind of create that what would be a naturally occurring antimicrobial envelope of propolis. But that smooth plank of wood on the inside doesn't really allow them to create that in a Langstroth hive, for example. Propolis also helps to regulate airflow, seals cracks, waterproofing, moisture control, and serves as the adhesion points for honeycomb, for the wax. A 60-gram envelope has been shown to positively affect colony level immunity to different pathogens. So, as I said, it may be helpful, especially if you don't have thousands of hives to do it with. Try to promote more propolis in your hive. Tremble dancing by resin foragers stimulates cementing bees, which are forager aged with atrophied wax glands, as well as middle-aged bees, and those remove propolis from the resin foragers and smooth it for use with their mandibles. So they have their work cut out for them. Diatonis bees, your winter bees, they have a low juvenile hormone level, a high vitellogenin level. They're immunocompetent and have an active fat body. They're long-lived and they focus on thermoregulation in the winter, brood care in mid to late winter, nursing, and then foraging. Some of those bees are 280 days old. So the emerging nurses that you might have seen today, if your weather was decent, they very well may be your foragers in April, and likely will be among other newly developed bees. So young, healthy diatonis bees are essential going into winter, which is why I stress adequate nutrition and ensuring your varroa mite levels have been well controlled at this point for at least the past month and a half or so. So vitellogenin is a glycolipoprotein which is biosynthesized in the fat bodies. It's intrinsic to the physiological plasticity of the bee, to immune function / disease resistance, and to juvenile hormone suppression. It's literally the fountain of youth for them. Starvation and freezing is also a leading cause of winter mortality. So it's essential to have ample accessible resources for a strong population. And in the Piedmont of North Carolina, I've always used the rule of thumb that 60 to 70 pounds of honey on top should get you through the winter. I've 65,000 cells full of honey, or sugar water or whatever, is essentially 65 pounds. So your colony's survival over winter requires a viable quality queen producing a healthy, sizable population of diatonis bees and the resources to support that undertaking, as well as proactive mite, disease, and pest control well before the end of brood production in your region and area, and timed to enable the best achievement of the goal. At this point, as we get towards the latter half of the middle of October, really just ensuring that they have the carbohydrates that they need. And I again would advise, if you can, cut a banana lengthwise, leave the peel on, and when you feed them the syrup, throw that banana in there also, and you'll have longer lived bees and healthier bees. And there's some great literature on that too, on bananas, which I dove into quite a bit, and so I keep bringing it up. But I do highly recommend it. It came as news to me not so long ago, so I went down that rabbit hole pretty deeply. But really, the timing is everything, as with all things bees. I just wanted to share this graph to show that is from Tom Seeley's The Wisdom of the Hive, how the weight differentiation at different times when a swarm leaves, and the timing of both providing resources, pest control, mite treatments, as well as swarm control, can really play a huge part in ensuring that you have a viable hive that will overwinter well. Also wanted to share this, thanks to Randy Oliver for letting me use it. It's in Manitoba in Canada, but it just provides a really interesting graph of where the turnover events happen. So roughly mid-October, the bees have all switched to their winter bee physiology, diatonomous bees. But as I said earlier, those were eggs that were laid even towards the tail end, middle to tail end of August through September. You'll still see some eggs now also, but I'm sure you can all attest that you saw a lot more in middle end of August and beginning of September than you're going to be seeing in the next couple of weeks. And then here are some of the references, and here are the rest of them that I used to put this presentation together for you. And I just wanted to thank you all for your kind attention, and I'm happy to take any questions. If you just typed in the bee supply in a Google search, or you should be able to find pretty easily that webinar too, which has a lot of the same information. But that's when I learned about the bananas also.
Okay. Thank you so much. You have a good evening.
Thank you. You too. Bye-bye. Thanks, everyone.
I think I heard him say.